For centuries, humans have selectively bred plants and animals to enhance desirable traits. We've sculpted crops for higher yields and nurtured animals for specific characteristics. Now, imagine having the power to edit the very building blocks of life itself, the genetic code. This is the promise of CRISPR, a revolutionary tool that has taken the world of biology by storm. CRISPR allows scientists to make precise changes to DNA sequences, akin to editing text in a document. This technology has unlocked unprecedented possibilities for understanding and treating diseases, developing new agricultural solutions, and even addressing fundamental questions about the nature of life itself. The implications of CRISPR are far-reaching, touching upon fields as diverse as medicine, agriculture, and environmental science. CRISPR's significance in biology cannot be overstated. It has democratized gene editing, making it faster, cheaper, and more accessible than ever before. Prior to CRISPR, manipulating genes was a laborious and expensive endeavor, often with unpredictable results. CRISPR has changed the game, offering a level of precision and efficiency that was previously unimaginable. This newfound ability to edit genes with such accuracy has ushered in a new era of biological research. Scientists can now study the function of specific genes with unprecedented ease, unraveling the complexities of biological systems. The potential for understanding and treating diseases is immense, as CRISPR offers a way to correct genetic defects at their source. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. While the name might sound like a mouthful, it essentially describes the unique DNA sequences that form the basis of this technology. These repeating sequences were first discovered in bacteria, where they serve as a sort of immune system, protecting against invading viruses. Scientists realized that this bacterial defense mechanism could be harnessed as a powerful tool for gene editing. By guiding a specific enzyme, often Cas9, to a target DNA sequence, CRISPR allows for the precise cutting and pasting of genetic material. This simple yet elegant system has revolutionized the field of gene editing. Section 4, CRISPR in Action, a step-by-step guide. The beauty of CRISPR lies in its simplicity. Think of it like this. You have a sentence with a typo, and you want to correct it. CRISPR acts like your editing tools, allowing you to find the error and replace it with the correct letters. First, scientists identify the specific DNA sequence they want to target, much like finding the typo in the sentence. Then, they create a guide RNA, a short piece of RNA that matches the target DNA sequence. This guide RNA acts like a GPS, leading the Cas9 enzyme, which acts like molecular scissors, to the exact location in the DNA. Once the Cas9 enzyme reaches the target site, it makes a cut in the DNA strand. This cut triggers the cell's natural repair mechanisms, which can be exploited to either disrupt the original gene or insert a new piece of DNA. The result is a precise modification of the genetic code. Section 5, CRISPR versus the old guard of gene editing. Before CRISPR, scientists relied on older gene editing techniques that were often cumbersome, time-consuming, and expensive. These techniques, such as zinc finger nucleuses and talons, relied on complex protein engineering to target specific DNA sequences. While effective in some cases, these methods were often inefficient and difficult to implement. CRISPR, on the other hand, offers a simpler and more elegant solution. The use of guide RNA to direct the Cas9 enzyme allows for greater precision and flexibility. Furthermore, CRISPR is significantly faster and cheaper than older gene editing techniques, making it accessible to a wider range of researchers. This ease of use and affordability have democratized gene editing, leading to an explosion of research in the field. CRISPR has become an indispensable tool for scientists worldwide, accelerating the pace of discovery and innovation. Section 6: CRISPR's impact real-world applications. The potential applications of CRISPR are vast and far-reaching, impacting fields as diverse as medicine, agriculture, and environmental science. In medicine, CRISPR holds the promise of curing genetic diseases that were once considered incurable. Scientists are exploring its use in treating conditions like cystic fibrosis, sickle cell anemia, and Huntington's disease. Beyond treating inherited disorders, CRISPR is being investigated for its potential in fighting cancer, developing new antiviral therapies, and even engineering organs for transplantation. In agriculture, CRISPR could lead to crops that are more resistant to disease, pests, and harsh environmental conditions, addressing global food security concerns. The revolutionary potential of CRISPR extends beyond human health and agriculture. Scientists are exploring its use in developing sustainable biofuels, engineering microbes for bioremediation, and even controlling populations of disease-carrying insects. The possibilities seem endless, limited only by our imagination and ethical considerations.